Please subscribe the channel and don't forget to click the bell icon.
Bismillahirrahmanirrahim. Welcome to Botany. So previously, in the previous class, we have discussed about the structure of the phloem tissue. What are the different types of cells which are present in the phloem tissue? What is the structure of the sieve tube elements or sieve tube member? What are the companion cells and how they are associated with one another? Fibers as well. So that is the simple overview, or we can say that a general structure of the phloem tissue. We have not discussed about the anatomy and the more about it, but we have discussed a general overview, a general structure of the tissue. And now we are going to discuss in today's video that what is the composition of the phloem exudate, or we can say that what is the composition of the sap, or what is the composition of the food which is transported from source to sink, what is the photoassimilates, or what are the types of molecules which are used for long distance transport in plants. So as far as this question is concerned, it can be answered, it may be answered by analyzing the chemical composition of phloem exudate. So it is very simple, or it is easy to tell the composition or chemical composition of anything by analyzing the chemical structure or the composition of that particular chemical. And phloem sap, which is flowing from the source to the sink, it can be collected from the aphid stylet, what we have discussed in the history class, the topic what we have discussed when we were discussing about the history, or alternatively from some plants by simply making an incision into the bark. So either we are using the aphid stylet, the aphid which is feeding on the plant, the stylet is incision is done in the stylet and exudate can be collected, or alternatively in direct incision can be made in the stem of any of the plant, and that cell sap which is collected, it can be examined, or composition can be. So as might be expected, the chemical composition of the phloem exudate is highly variable. So it is seen in the various plants and in the various species that the composition of the phloem exudate, what is the composition, what is the percentage of different types of molecules in that phloem exudate is variable from species to species, from genus to genus, from plant to plant, or we can say that from individual to individual. And these composition of the phloem exudate not only differs from species to species, it also differs according to the situation the plant is facing. So as far as the phloem exudate is concerned, it is not the case that the composition is fixed, and whenever it is transported, so it will be the fixed composition. So it is not the case. So the composition of the phloem exudate depends upon the nature of the species, the species type, the individual type, the habitat of that particular individual. It is also possible that some plants of the species which are occurring in different types of habitat, they have a different type of exudate which is flowing or which is moving. So it is difficult for us to make a generalization that this is the phloem composition of phloem exudate that is transported in all the plants. So it is not the case. So it depends on the species, and as we have discussed, that it depends upon the species, and in the species it is also different from the individual to individual, and it depends upon the habitat in which the plant or individual is living. The age of the plant, younger plant or the seedling, younger plant have a different composition of the phloem exudate, and those plants which are mature and which are old age, or we can say that which are they have a different type of composition and physiological condition. And what are the physiological condition the plant is facing of the tissue sample? So even for a particular sample under uniform condition, there may be wide variations in the concentration of particular components between subsequent samples. So even the conditions are uniform, conditions are the same, but the composition may differ, as we are discussing in the next discussion, that the composition of the phloem is different, or it is different for species to species, and it differs from one sample to another. Why? Because There are also other some compounds like nitrogenous compounds and hormones as well, which are transported along with the food molecules, and its concentration differs from one physiological condition to another. So for example, we have analysis of the phloem exudates from the resinous communis and that is shows that the exudate contains the sugars. They also contain the proteins, amino acids, the organic acid malate, and a variety of inorganic anions and cations. So sugars, in the sugars we have raffinose, stachyose, and most probably and most abundant, that is the sucrose, which is translocated from source to the sink. We have also different types of proteins which are translocated. We have amino acids, certain amino acids which are transported along with the organic acid malate, and you have discussed in the different types of photosynthesis which is occurring in the plants. For example, you have discussed about the CAM, we have discussed about the C4 photosynthesis and CAM photosynthesis, where the organic acid malate has a very key role in that particular pathway, and there is also certain inorganic anions and cations as well. So as you can see, there is not simple sugars which are transported through the phloem or through the phloem tissue. There are also some other compounds. There are proteins, amino acid, organic acid, and most probably the composition differs in these. These compounds have different concentration, different percentage of the phloem exudate. So predominant amino acids as which are transported from phloem are sourced to sink. They are glutamic and aspartic acid, which are common forms for the translocation of assimilated nitrogen. So whenever the nitrogen is assimilated into a protein or into an amino acid, mostly it is assimilated in the form of glutamic or aspartic acid, and then these two, glutamic and aspartic acid, are transported along with the sugars to the lower parts of the plant where they are utilized. So inorganic anions include, so as far as the inorganic anions are concerned, they are phosphate, sulfate, chloride nitrate is conspicuously absent. So inorganic ions, either they are phosphates or they are sulfate, but chloride and nitrate, they are conspicuously absent, while the predominant cation is potassium. So nitrate and nitrite, so they have a role in the nitrogen fixation when we discuss about the nitrogen fixation, nitrogen cycle. So some plant hormones along with that particular food molecule, they are also transported, like the auxins, cytokinins, the gibberellins, they were also detected, but they are in very minute amount, as hormones are also in always in very minute amount they are produced, but they are used to stimulate or used to initiate a response. So protein found in phloem exudates include a wide variety of enzymes as well as one predominant protein called the P protein, so that is unique to the translocating cell. So as far as the transport in the phloem is concerned, the P protein has a very important role, which we are going to discuss in the further discussion. So along with the sugars and other compounds, there is also certain enzymes which are also transported and protein. So by discussing these, so there is a composition, different composition in different species. We have discussed about this. There is a sugars which are transported along with the proteins, the amino acids, the glutamic malate, the different amino acids, glutamic and aspartic acid, along with the anions, cations, along with the auxin, cytokinin and gibberellin. So mostly the food molecule which is transported, that is used for the respiration, that is the sucrose in plants, but the composition always differs here in the flow in the other parts, like there may be a different concentration of hormones, different concentration of amino acid, which will largely affect the composition, overall composition of the phloem exudate. So this table shows the chemical composition of the phloem exudate from resinous communis. So as you can see from that, sucrose has 80 to 106 milligram, that is the milligram per liter. So predominant sugar which is transported, predominant molecule is the sucrose, which is transported, and its concentration is higher, far more higher than any other compound, that is it is 80 to 106 milligram per liter. So as far as the proteins are concerned, they are 1.45 to 2.20. There is the amino acids which are 5.2, milligram per liter, or malic acid 2 to 3.2 milligram. So either along with the organic molecules, there are also inorganic compounds which are milli-equivalent, and they are anions like 20 to 30%, cations 74 to 130%, and as far as their total dry matter is concerned, percentage is concerned, they are 100 to 125 milligram per liter. So this is the overall composition of the phloem exudate from Ricinus communis. So this is another source. As you can see, this is the milligram per liter. The sugars, Ricinus communis, there is 80 to 106%, 106 milligram amino acid. We have 5.2. Organic acids have 2.0 to 3.2%, along with the potassium, protein, potassium chloride, phosphate, magnesium, and other compounds. So this is the overall composition of the phloem exudate, the food molecule, the food which is transported in the, or the sap which is transported in the phloem tissues. So the principal constituents of phloem exudate in most species is sugar, and in sugar, when you are discussing sugar, the sugar has a diversity of compounds, and in castor bean it is sucrose which comprises approximately 80% of the dry matter, and not in only the castor bean, in majority of the plants, sucrose is the predominant molecule which is transported as a food molecule from source to sink. So, but there is also some exceptions to every rule in every thing in the science. When we are discussing, there are also some exceptions. So there is a small number of families in the angiosperm, they translocate in addition to sucrose. So there is a sucrose. So it is not the case that only sucrose will not be used. Sucrose, oligosaccharides of the raffinose series, raffinose, stachyose, or verbascose. So these are all the compounds, or we can say that these are all the derivatives. They are all sugars, but these are also transported in addition to sucrose. So along with the sucrose, there is also some compounds, sugar compounds, which are transported. So stachyose, for example, accounts for about 46% of the sugar in stem internodes of cucurbita maxima. So as far as the stachyose is concerned, it is about 46% in the cucurbita. And yet another families like the Oleaceae and Rosaceae translocate some of their photostimulates as sugar alcohol, like mannitol or sorbitol. So these are the exceptions. So when we are discussing something in the botany or in any other field, so we talk about the generalization, we talk about which is the usual process or usual composition. So usually the sucrose is the major transport compound, transport molecule, which is transported from source to sink. So these are all exceptions. There may be other exceptions as well, but these are certain exceptions which we know. So there is another question, that why sucrose is, or why the food is transported in the form of a sucrose and why not glucose or any other sugar molecule. So that is one possibility, is that the sucrose is a disaccharide. So we know the disaccharide, but in disaccharide we have a lot of compounds, diversity of sugars which are all disaccharides. But and its related oligosaccharides are non-reducing sugars. So that is a disaccharide, and along with the sucrose, and not only the sucrose, its related disaccharides, other they are all non-reducing sugars. So on the other hand, all monosaccharides, including glucose and the fructose, they are all the reducing sugars. So what is why we are talking here about the reducing and non-reducing sugars? Because in the reducing sugar there is always a free aldehyde or a ketone group which can subsequently or which can easily be oxidized by the different compounds which are present in the cell. So sucrose molecule has to pass through the cytoplasm of the phloem tissue or the seed tube elements, and it has to pass other cells. So the food molecule should be in the form that it cannot be degraded, or it cannot react with other compounds, so that its chemistry and its nature should not be changed. So that's why the food molecule is transported in the form that is inert in the form, or it cannot, or it has very less reactive in nature. So reducing sugars have a free aldehyde or ketone group, and that free aldehyde or ketone group which is present in the aldehydes and the ketones, they are capable of reducing mild oxidizing agents. So they are able to, or we can say that they are able to, or they are very much reactive, and they can react with other compound and which will affect the process of long distance transport. So some oligosaccharides such as sucrose along with other compounds, they are non-reducing sugars because the acetyl link, what is acetyl link? The linkage between the glucose and the fructose, which will result in the sucrose, that in the subunit is stable, and that linkage between the two molecules, between the two subunits is stable, which will result in the inert molecule, and which result in the non-reactive molecule in the alkaline solution. So the exclusive use of non-reducing sugar in the translocation of photostimulates may be related to this greater chemical stability. So use of this sucrose molecule as a source of or as a transportation material, as a transportation vehicle for the plant, so it may be attributed to this particular ability. So non-reducing sugars are less likely to react with other substances, like the glucose is reactive and it can react with other compounds in the pathway to which it is transported. Indeed, glucose and fructose, they are both reducing sugars, are rarely found in phloem exudates. So as glucose and fructose, when they are in free form, they are very much reactive, so they are not found as far as the phloem exudates are concerned. But when the glucose and fructose, they join through the linkage, glycosidic linkage, so what will be the result? They are making the sucrose and it will make it inert. So here we have the reducing sugars which are not generally translocated in the phloem. So why they are not translocated? As you can see in the D-glucose and D, here the D means dextrorotatory or levorotatory glucose. So that is the stereochemistry of the glucose molecules or the mannose molecules or the sugars which we have studied in the biochemistry. So that is dextrorotatory glucose, D-glucose, it contains the free aldehyde group along with the D-mannose, which is also contains the aldehyde group, which can react or which can have the power to reduce the oxidizing, mild oxidizing agents. So there is a fructose, it also contains the free ketone group. So when these glucose and fructose, they are joined in a way, so they result in a structure, they result in a form that can be transported in the relatively inert form.